sequencing on different tissues in the body and identify relationships between genotype and expression of various genes. There are limitations to these approaches, however, that have started to hinder our progress as a field. So, for example, many of these lack temporal and cellular resolution. So GWAS, of course, doesn't really tell you much about individual cell types that are associated. The EQTL detection studies do, to a certain degree, through bulk RNA-seq. There are approaches now, so single cell based EQTL detection studies that are starting to pick up pace that can help with some of the cellular resolution. But for the most part, this doesn't really tell us a lot about which organ systems or tissues are being impacted, or at least that are associated with that genetic risk. Often you need hundreds to thousands of samples, again, to dissect complex traits. It seems like the more samples you have, the more loci you're powered to detect. Also, it's very hard to quantify the contribution of environmental factors, viruses, air pollution, what have you. Often we use proxy measures for this. So for air pollution, one of the things that groups tend to do is look at where you live and infer from there the levels of exposure that you have the levels of exposure over the course of your life. So again, it's hard to quantify this, and sometimes this leads to some of the genetic signal being over quantified or over represented. Again, we have minimal insight into mechanism when we use these approaches. And though this is something that's being corrected in the field, for the most part, these studies are dominated by European subjects. So not a lot of ancestral diversity in these approaches thus far. Now, human in vitro models can overcome some of these issues. I don't need to explain this too much to this group. This idea that you could take stem cells and convert them to many different cell types of interest. Now, what this has allowed us to do through a range of technologies that have been developed over time is start to identify the function of some of these disease-associated variants. So we can look at EQTLs, as I mentioned, both context and cell type and staged specific EQTLs. We can perform these sort of EMPERA or CRISPR screens to interrogate the function of some of these genes. And you can do this in a range of different human cell types. My lab and many labs here are interested in neurons and neural cell types and other sort of cerebral or brain type organoids. So the value of this is you can start to interrogate some of these candidate variants with high cellular and temporal resolution. However, again, there are still some drawbacks to this system. First of all, many of these studies are attempts at trying to interrogate the function of variants that have been identified from some of these large-scale GWAS studies. The problem is the signal is often abundant, so there's many loci associated, and it's often quite noisy, meaning there's many variants within a loci that are associating with the trait or the disease. An example here is one of the 108 hits from this schizophrenia GWAS published in 2014. So that can be often difficult. There are a lot of fine mapping tools people use in order to identify or at least nominate one or a handful of variants within these loci. But again, if you're basing your study off of this, it can be hard to try to figure out which variant you want to focus on for in vitro work. Now, if you want to do variant discovery in vitro, that's also quite challenging. Typically, this is done in an array format where you have cell lines cultured separately from each other to avoid donor mixing. The problem is often these are underpowered due to the limitations in the cost and complexity of culturing all these cell lines for prolonged periods of time. By having them cultured separately, this introduces its own technical noise. So the well-to-well, batch-to-batch, plate-to-plate variation can often dominate some of your signal and mask biologically important signal. And again, most of these studies have been performed on European lines, though again, I do think the field has acknowledged this limitation, is doing a pretty good job of trying to address this. A lot of the biobanks, for example, are focusing on generating more iPSC lines from individuals from diverse ancestries, which is very important. So as a postdoc in Kevin Eggen's group that was at Harvard at the time, that now the lab shut down in 2021 or 2022 because Kevin moved to BioMarin, which is a company out here in California. But prior to that, we had tried to address this question by saying, we really need a high throughput platform for these sort of population scale investigations of these risk variants in human cells. And so what we did is we took all these cells. So this is actually an actual picture from our incubator at the time. We took all of these different cell lines that are usually cultured separately, and we put them all into the same dish. Excuse me. And this is what we're calling a cell village. And so this is population genetics in a dish. You can take male and female human donors representing different ancestries, different disease states, whatever is most important to your biological question. You can take these cell lines from different populations or different members of the population and culture them all together. And one of the really nice benefits of this system is that once they are all together and they now have a uniform environment, you can then introduce a range of different cell extrinsic factors and ask questions about response to these factors and how genetics can influence that response, as well as start to integrate some of the mechanisms that are driving that differential response. So a lot of things we can do here. I know some of the past presenters of the seminar series have also highlighted some of these benefits to the village system in terms of interrogating response to environmental perturbations. So my lab is focused on using these villages to understand early brain development. We're very interested in this, particularly in neural progenitor cells, because we know that disruption of the activity of these cells, whether it be proliferation, differentiation, what have you, can have lifelong consequences. Many neurodevelopmental disorders like autism and intellectual disability are all associated, or many of those diseases are associated with the activity of these cells. These cells are present very early in development, and for the most part, these fetal early neuroprogenitor cells are gone by the time we are born. There are some disease models in animals, particularly in mice, in which the disease mechanism appears to be that these progenitor cells tend to still be present after birth. But again, for the most part, we are studying very early fetal brain development when we're focusing on this particular cell type. So a brief overview. Again, this is work that was published in Cell Stem Cell in 2023. And I'll stick again pretty closely to what we talked about there. First, I'll talk about our accelerated and robust protocol for generating these neuroprogenitor cells from many, many different human stem cells. Really, the idea here is if you want to make a village of this cell type, you have to make sure your induction protocol is working consistently. Next, I'll show you how we generate and deconvolve these mixed donor cultures. We'll give you some insight into our construction strategy. And then finally, we'll give a story about how we use this village to identify genetic risk for Zika virus infection of these prenatal neuroprogenitor cells. So first, a bit of a history lesson as to what got us close to this point. This is all founded on 15, 20 years of work that started in the early 2000s that really started to find ways to generate neuroprogenitor cells and other neural cell types from human pluripotent stem cells. Now, initially, this work was done in embryonic stem cells and then subsequently in induced pluripotent stem cells. But the two main protocols for this involved either generating embryoid bodies and then plating these neuroprogenitor cell mixtures on an adherent surface, and then either manually or enzymatically or even using FAC sorting to pick out your neuroprogenitor cells of interest. And this is reported to take anywhere from 16 to 50 days per cell line. And a little bit later, the Studer Lab published this dual SMAT approach, which enables you to make these neuroprogenitor cells by plating them at high density and then using patterning factors. And then within 11 to 17 days, you have a nice confluent bed of pure neuroprogenitor cells. So there are many advantages to these systems. First of all, they tend to generate these homogeneous cultures. There's a certain level of developmental fidelity to them, meaning that they mimic some of the early induction steps that take place in the human brain. And they've been functionally validated left and right, again, for 15 to 20 years in some cases. However, they do, some of these require selection, especially for the embryoid body system. The dual SMAD protocol is density dependent. For any of you who've worked with this, you may notice that from line to line, the number of cells you start with actually could influence dramatically the overall outcome of the culture. And also, there's a significant amount of batch to batch and cell line variability that we observed in the Eggen lab, as well as others have reported in the literature. So we really felt like in order to make neuroprogenitor cell villages, we really needed a new way of making these cell types. So what we did was we decided to leverage existing protocols for making postmitotic neurons. So the idea here is that we introduce neurogenin 2 using lentiviruses into our human pluripotent stem cells. And then we can start induction by adding doxycycline. We also include some of the same patterning factors used in the dual SMAD protocol. And we do this for 2 days. And we found that that is sufficient to generate these neuroprogenitor cells. We also have a selection step with puromycin to get rid of any cell that didn't have the lentivirus in it. So we're calling this stem cell-derived NGN2 accelerated progenitors, or SNAPs. And really, this allows us to do the induction in 2 days, followed by expansion from that point on. I will mention at this point that we are working on a 2.0 version of this that does not use lentiviruses. It uses AAVS1 safe harbor. We target NGN2 to the AAVS1 safe harbor locus. I don't have much data on that yet, just beyond some immunostaining. But we do think in the next 2 or 3 months, we will have a lot of data supporting that method. It does appear that we are going to switch entirely to this new method that I don't have data on quite yet. But if there's any questions about that, more than happy to answer them. Here's some validation of the SNAPs protocol, the one that we published. We see that these cells express canonical protein and transcriptional markers. So we're seeing nestin, PAX6, SOX1. We don't see any OCT4, which is the marker for the pluripotent stem cells. So we do feel like we are doing a good job of getting rid of those contaminating cell types. When we looked at these cells using single-cell RNA-seq and compared them to in vivo data sets and asked what cell type do they most closely resemble, overwhelmingly these cells resemble these early radioglial-like cells, which in this data set from the Nowakowski group, paper from Science, that was the earliest neuroprogenitor cell type they had represented. For the most part, we are seeing these cells mimic these early neuroepithelial cells. And it looks like I'm missing a bulk RNA-seq data that I had in this slide that somehow disappeared. But our bulk RNA-seq data is also supporting that, and our bulk RNA-seq data suggesting we're making dorsal telencephalic NPCs. You also see that this production of dorsal telencephalic NPCs is very robust. So when we use just the NPC marker assay, looking at immunostaining, we reported that 111 out of 115 stem cell lines were successful on their first try. Success was denoted at this time as over 75% positivity for the positive markers for nestin, PAX6, and SOX, but below 1% for OCT4. Again, I'll mention that we are getting much higher purity for the 2.0 version of this, but again, don't have a lot of data for that quite yet. We also ran the multipotency assay, which has shown that these cells functionally behave as neuroprogenitor cells, meaning they can become both neurons and glia upon spontaneous differentiation. And we tested this in 30 of our SNAP lines, and all 30 passed this test. Again, hinting at the reproducibility of this effect, we did single-cell RNA sequencing on, I think, 40 lines are represented here. And we quantified here what cell type they most closely resembled, again, using that Nowakowski data set as our reference from in vivo fetal brain. And as you can see in gray, a vast majority of these cells are identifying as, or most closely resemble these fetal neuroprogenitor cells. Excuse me. So based on that, we felt like we had sufficient data to suggest that we could start building villages of neuroprogenitor cells for some of these questions that were interesting to us. Now, what I'm going to show you here is some work showing how we make these villages and how we can actually use different bioinformatic tools to answer biological questions using them. So this is a video from my lab. So again, most of what I'm going to talk about today is work done from my postdoc. But some of I just want to give you some insight as to what we're doing in our lab. This is our entire tissue culture space currently. So it's pretty small. We have 2 hoods in there, and that's pretty much all we need right now for what we do. We are working with a 300 donor stem cell biobank at this time. We'll probably get another 50 or 60 next year to really increase the ancestral diversity is one of our main goals currently. As I'll show you here, we induce in an array. So each cell line is generated as NPCs separately. We do some of the QC I just showed you, and then we freeze them down for later construction. On the day of construction, it requires a 3 to 4 person team, 2 of which are pretty much in the hood, and the other 2 are sort of roaming around helping out, whether it be with counting or with finding reagents or media, whatever it may be. We found it's very helpful to have those individuals outside the hoods to help. Right now, I should say that at the time of our publication, counting was the rate limiting step. So whenever you're passaging a bunch of cell lines to pull them together, the counting part can take forever. And there's an asterisk, and I'll explain that asterisk in just a second. So for example, for a 20 donor village, it took us over an hour just to count those 20 donors using our automated countess. So you can see them kind of in the back. We have 2 countesses for automated counting, but even then, it was taking over an hour. For our 45 donor village, it was taking 2 hours for counting, 3 hours total for the whole construction. Now, I put an asterisk there because in the last month or 2, we have developed a new way of counting multiple lines simultaneously that uses a automated microscope that can look at 20. It's all plate-based now, so we're counting 24 samples in about 3 minutes. So it's dramatically reduced this step, which we think is going to be very helpful for building larger villages, at least until we get some robotics in here that can build villages from scratch. We think that this is going to be a really important improvement on the protocol that will allow us to get to a much bigger donor sample sizes. So here's how we actually can start to answer questions using this village system. So we take cell lines from multiple different individuals. Each of these cell lines have been genotyped in some way. So some of them have been whole genome sequenced, some of them have been SNP profiled, and then the whole genome is imputed from there. Jim Nemish at the Broad Institute, who developed these bioinformatic tools, has found that it doesn't matter if it's whole genome sequenced or SNP imputed, if SNP imputed, the call rate, the accuracy of calls for downstream analysis are equally accurate. So we're talking about above 99.9%. That's important because whole genome sequencing is a lot more expensive than SNP profiling. And so we found that it's a lot cheaper to just do the SNP profiling of each donor and then impute from there. So as I mentioned, we do our inductions separately. So each line is cultured separately for induction. And then when we're ready, we pull all the cells together into our village. Now we have 2 bioinformatic tools that we use quite heavily. One is called dropulation, which is based on single cell RNA sequencing of the cells in the village. Now, what this does is it uses their genome as the barcode, right? We already genotyped everybody. So there's a natural barcode in these transcripts. So there's many variable SNPs in these transcripts that we can use to re-identify donors. So what that allows us to do is create donor level gene expression matrices. So for every donor you have in the village, you can generate a separate gene expression matrix. So obviously we can use this for a range of different things. You do differential gene expression analysis, WGCNA. I'll show a little bit of data here in the next slide or 2 looking at expression quantitative trait loci detection using the dropulation system. Now we can also do high throughput cellular phenotyping using a tool called SensaSeq, which is based on low coverage whole genome sequencing. By low coverage, we're talking 0.5x, 1x in that range. This currently costs us like $60 to $100 a sample, depending on who's doing it for us. So it's a really nice low-cost way of generating a lot of phenotyping data from your village. What this does is it essentially does what its name implies. It runs a census on who's present. It's saying who's in the village and what proportion of the village they represent. So if you have 100 donors in your village and you did a really good job of counting them when you put them all in, Census-Seek should tell you that each donor is 1% of the village. And of course, that can change over time, and we can actually use those changes over time as a phenotypic measure, which I'll explain in a couple slides here. Okay, let's talk a little bit about dropulation, some of the stuff we can get. This is some of the data that we reported in the paper. What we did here is we had a village of 44 neuroprogenitor cell lines. These were all embryonic stem cell derived, and these are all European ancestry for this work. Now, what we did is we used the genotypes that we had. In this case, these were all whole genome sequenced. And we used our donor-level gene expression matrices to identify, in this case, a little over 24,000 EQTLs in roughly under 1,000 genes. So I want to highlight a couple things for the developmental neurobiologists in the room, just really highlighting that not only can you identify these, but it could potentially be a good source of hypothesis generation. So MAPK3, TRK1, DMPK, these are all genes that play really important roles in early brain development. And we know that they're highly expressed in neuroprogenitor cells. So when you see this sort of natural variation across our donors in expression, and it appears to be associated with specific variants, you can design experiments around these ideas and ask questions about, say, how your genotype at this MAPK3 loci can influence, say, neuroprogenitor cell proliferation or differentiation. So you really want to highlight that. And I think that's one thing that I really hope to get out of this group as a whole, that we really start to think about how we can leverage each other's data sets to answer important questions in our respective fields. The other thing I want to mention is we can also cross-reference these EQTLs to in vivo data and ask questions about how what we're seeing in a dish may correspond to what's happening in living human populations. So one I want to highlight here is one of the strong EQTLs we detected in our village of neuroprogenitor cells was actually one of the top hits in a cortical surface area GWAS that was published in Science in 2021. And what we see here is that one of these genes is encodes this, what is this, link 0221O. This is a protein that shares sequence identity with corticotropin-releasing hormone 1. Now, what's interesting to us is cortical surface area. If you're a follower of the radial unit theory, one of the driving forces behind cortical surface area is the proliferation of early neuroprogenitor cells. So again, this could be an example where we look at differential growth rates across these three different genotypes and ask if there's a functional link between these variants in this GWAS hit as it corresponds to cortical surface area. So again, something we hope to either do ourselves in the future or work with others, or to sort of start to answer these questions and bridge these gaps between in vitro and in vivo work. A little highlight here on Census-Seek, how we can do rapid cellular phenotyping using this tool. As I mentioned, one of the things you can simply do is just ask how donor proportions change over time, whether it be just under static conditions or after a treatment of some sort. In this case, we have some interferon gamma data showing that for some donors, they dramatically increase their proportion after treatment, and others it's the opposite effect. Now, we really haven't done much with this data thus far. We are working with Harold Pimentel, who's a bioinformatician here at UCLA. We're developing a lot of models on how to properly interpret Census-Seek data, mainly because we have to take into account the ratiometric nature of this data. And so some of the more streamlined ways you might do this might be violating certain statistical principles. And so we're really close to having some really good answers on how to address this. And we hope to make that available in the near term. Another thing you can do, and I'll give examples of this in a few slides when we talk about our Zika story, is you can fact sort the cells in your village based on expression of different markers. And then once you do that, you can run Census-Seek on the different fractions and calculate what we're calling an abundance ratio, which is the donor's relative representation in the two fractions. This can actually serve as a quantitative phenotype. And as I mentioned, we'll show you how we did this with Zika virus infectivity and use this to rapidly quantify infectivity rates within a village across individuals. One of the things I'm most excited about is this ability to, from a couple of different wells, from the same batch of cells, you can cut across multiple levels of biology. So you can take your village, you can subject some of the cells for census-seq analysis, some for single-cell RNA sequencing, and then integrate that data later on with either GWAS or TWAS or WGCNA data. We're working on various projects that leverage this sort of integration of the different data sets. And then, of course, after you find either individual variants or disease mechanisms or genes of interest, we recommend validating them with either individual CRISPR tools or pharmacological modulators, so that we can validate that what we're seeing is not being driven by sort of cell non-autonomous factors, but instead by true cell intrinsic behaviors. And I'll have a little bit of data of that later on when we talk about Zika and whether or not what we're seeing is actually intrinsic to the donors. Okay, as promised, we have our story on how we use this to identify genetic risk for Zika virus infection of neuroprogenitor cells. So for those of you who may be unaware, at the time in 2016, Zika was considered an emerging global threat. I still think it's considered an emerging global threat. It's just, of course, not the virus on top of mind for everybody since the COVID pandemic started. But regardless, we're very interested in this virus because it's transmitted by mosquitoes. So it's in the same family as dengue and yellow fever. It's characterized by these periodic outbreaks, the most famous of which took place in northern Brazil in 2015, 2016, that moved into Central America and into the southern portions of the United States. I became quite interested in this because I'm Salvadoran, and this virus is actually starting to affect a majority of my family, which still lives in El Salvador. They are being exposed to this virus, a couple of whom were trying to get pregnant at the time. And the reason that's important is because most people don't show symptoms. But if you are a pregnant woman and you're exposed to this virus, you are 17 times more likely to give birth to a child with a birth defect. The most famous of which is microcephaly, or small brain or small head. But we've since learned that even if the child does not present with any overt, obvious birth defects, they're still at a much higher risk for other developmental conditions, autism being one of them. A lot of sensory and motor issues have also been identified in these children later in life that indicate that the fetal brain is being significantly impacted by exposure to this virus. Now, some of the earliest work on Zika found that it tends to preferentially target proliferating progenitors in the prenatal brain. We validated some of this work as well, showing in both neuroprogenitor cells and post-mitotic neurons that the virus tends to hit these progenitor cells and not so much our post-mitotic neuron models. So one of the first questions we asked, this is before we even had villages up and running, was are there differences across individuals in their susceptibility to infection? So we had an array culture of 24 lines. So these are cultured separately. And we hit them all with the same amount of virus at the same time and fixed them all at the same time. And we just did immunostaining to quantify infectivity. And as you can see here, there's a huge range in susceptibility. Some lines were not really infected at all. We're talking 0.7% of them were infected. Others, like MEL2, were above 99% of the cells were infected. You see this is nice gradient here in terms of infectability. So we asked ourselves, okay, well, we're seeing this cellular phenotypic variation. Is there any genetic or molecular variation that can explain what we're seeing here? And so that spawned some of this early work to explain this question. So we first realized at the time that with our SNAP model, we should probably make sure that we know which genes in the human genomes, what are the host genes that are involved in the Zika virus life cycle. So which genes are potentially contributing to whether or not these cells get infected or whether or not they die in the long run. So what we did here was a genome-wide CRISPR-Cas9 screen in which we hit our neuroprogenitor cell model, our SNAP model, with either mock media or one of two different Zika strains, either the Uganda strain, which is the original from 1947, or a strain from Puerto Rico that was isolated in 2015. We then gave them 10 days, and we asked which cells survived, and then What we did is after those 10 days, we harvested the cells, did DNA extraction, and asked, is there an enrichment of certain guides that would suggest that those genes contribute to the Zika virus life cycle? So what we're seeing here are, first, we're going to talk about the host factors. These are the genes that the virus likely hijacks in order to execute on its life cycle. So what we're seeing here are many genes that are in pathways that we expected. So heparin sulfate genes, for example, play a role in viral attachment, whereas the integrins were found to play a role in viral entry. Once the virus is inside, it tends to go to the ER membrane complex to create its replication factories, essentially. So what we're seeing here is if we get rid of any of those components, the cells tend to survive a little bit better, which is why they are enriched later on in life, or later on in the experiment. On the other side, we found a bunch of restriction factors. Now, these are the genes that presumably protect the cell from infection. Now, what we mean by that is if we got rid of these genes, they really did not do well. They were underrepresented in the final guide pool when we did the sequencing. So these include IFNAR1 and 2, STAT2, and a gene I want you to remember called IFIT-M3. What we next did is we took our list of E genes that I mentioned up front from our EQTL detection from our village of 44 donors. We then cross-referenced those genes with the 125 or so genes that we identified from the screen I just mentioned to you. Now, there's an overlap of 7 genes, and then we filtered by the effect size of the EQTL that we had detected, and there's a clear winner here. And it was this variant in the IFIT-M3 gene, or near the IFIT-M3 gene, I should say. The SNP is identified as RS344-81144. That will be important later on in this talk. So what we're seeing here is that donors that have the CC genotype have a much higher expression level of this IFIT-M3 gene than donors that have the TT genotype. And so this is very interesting to us if you look at the presumed function of this gene. As I mentioned, our screen identified it as a restriction factor, so it plays a protective role in the cell, and others have found similar things. So first of all, others have found that this specific variant is in the CTCF domain. It's in the core promoter of IFIT-M3. And so it's been previously shown that having a C allele at this site leads to higher expression, whereas the T tends to increase repression of expression. And so the T variant, or the T allele, is what seems to be driving down expression of this nearby gene. Now, there's a group that published a paper in 2019 really nicely elucidating the mechanism by which IFIT-M3 protects cells. So what it does, it's actually present in the endosome, in the membrane of the endosome. And it can detect certain enveloped RNA viruses. Now, when it detects those enveloped RNA viruses, it shuttles that whole endosome to the lysosome for degradation. Now, if you don't have IFIT-M3 in the membrane, or there's fewer copies of the protein floating around, well, the sentinel system is less effective. And it means that more of these endosomes are able to, more of the virus, I should say, within these endosomes are able to uncoat and attach to the ER and create their replication factories. So our hypothesis, based on our early data, as well as what we saw in the literature, was that the donors with the C genotype at this site were more likely to be protected from infection compared to those with the T, because they have higher expression levels of this IFIT-M3, at least at the transcript level. So we then decided to test this hypothesis formally, going back to that exact same village I had already described to you, where we infected with either mock media or Zika virus, who waited 2 days and in fact sorted the cells in the village based on whether or not they expressed the Zika virus envelope protein. So we fact sorted into positive and negative fractions, and then did census-seq on each of those fractions to calculate this abundance ratio that I mentioned earlier in the talk. So as you can see, our hypothesis was supported. Now, what we're showing here on the graph on the right is that the donors with the CC genotype have a negative abundance ratio, meaning they're more likely to be in that negative fraction compared to the positive fraction. Again, these are the donors that have higher levels of IFIT-M3. The TT donors were the opposite. They had a positive abundance ratio, more likely to be in the positive infected fraction compared to the negative uninfected fraction. You can see that the p-value and the r-squared suggests that in this case, it was like a little bit over 25% or 20% of the variation could be explained by this single variant in the genome. We then went back and asked, okay, is this being driven by something weird in the village? Is by mixing 44 different cell lines together, is that what's actually creating this hit, or is there something else, or is this actually truly intrinsic to these donors? And what you can see here is we took that exact same 24 donor arrayed experiment that I talked about a few slides ago, and all we did was group by genotype. So this is immunostaining base. You can see representative images there in the middle. And we're seeing here that, again, our hypothesis is supported. The donors with the CC genotype have much lower levels of Zika virus infectivity compared to the donors with the TT genotype. We then took 36 cell lines that were not included in any of these experiments and repeated this array-based experiment. And in this case, not only was our hypothesis supported, but the genotype at this single site could explain almost 60% of the variation across our donors in their Zika virus infectivity. We then took this data and ran a GWAS on it. And as you can see, these were the only two hits that reached genome-wide significance. It included our hit, our RS344811144, as well as a SNP that is in LD with our hit that is actually in an intronic region. These were both showing up. To be clear, though, it's pretty clear functionally that it's most likely that it's our IFIDM3 promoter SNP that is driving the biology. Okay, very, very important slide. Very, very important slide to further show you that what we're seeing in a village is intrinsic to these individual donors. We looked for correlation between our arrayed results and our village results. You could see a very high correlation between the two, again suggesting that we are capturing cell intrinsic properties here with our village system. Okay, last two slides. I just want to highlight something very interesting we noticed when we looked at the 1,000 Genomes Project data set, looking at the allele distribution, the allele frequency distribution across global populations. As you can see, that the risk allele, the T allele, is pretty much non-existent in certain populations in Japan, Southeast Asia, Africa, to a certain degree, Central and South America. But it's at a much higher proportion, much higher frequency in these European populations. You may also notice that this map is color-coded red and blue. The red actually corresponds to the terrain inhabited by these A. aegypti mosquitoes that carry this family of flaviviruses. So it's very interesting that where those viral vectors are located tends to be where this risk allele no longer, I shouldn't say no longer, where this risk allele does not exist. So it really introduces this question as to whether or not this has been a driving force of some sort of selective sweep in some of these populations over time, whether it be some of these viruses or a precursor to some of these viruses that had similar effects on overall fitness. This is important because there does appear to be this impending collision between these viral vectors and vulnerable populations. What I mean by that is because of climate change and increased globalization, these mosquitoes are expected to move further north into portions of the United States and Europe. And again, these are the areas where the populations there, the ancestral populations at the very least, tend to have higher proportions of this risk allele. And so it's possible that we're going to have more exacerbated outbreaks because of this lack of natural protection. On the flip side, though, perhaps we could use this information to identify those at-risk individuals or even develop therapeutics that are designed to enhance expression of IFIDM3 under baseline levels or at baseline conditions to help protect us from these infections. So as a little bit of insight into the future, we were recently awarded a CERN grant to expand this analysis, where we're building a village of 150 donors. This includes equal share of European, African American, and Hispanic Latino individuals. We're not only looking at infection of Zika, but other viruses like cytomegalovirus, for example. We're also asking questions about how our genetic background influences our response to cytokines and inflammation, which of course also play a very important role in the infection to disease pathway. So just as a closing thought, our cell villages capture genetic, molecular, and phenotypic heterogeneity in a shared, uniform in vitro environment. We use a dropulation tool to identify expression of quantitative trait loci, as well as Census-seq to do high-throughput cellular phenotyping in our villages. And I just shared the story using, which we found a SNP in the IFIDM3 promoter that appears to play an important role in differential susceptibility to Zika virus. So with that, I want to acknowledge my lab. here at UCLA that we launched in September 2021. We're fully devoted to villages and expanding the technology, expanding the tools we can use for this, expanding the questions we can answer for it. So again, really happy to be part of this group to hopefully find some similarly motivated individuals and who are equally committed to advancing this technology. So more than happy to answer any questions you may have.
Thanks, Michael.
Ooh, voice held up. That is the
Yeah,
remarkable
well
part
done. Any questions?
I have a question.
Yes, Sally.
Well, congratulations. How exciting this is, really. And my husband had dengue, so again, in Haiti. So I can tell you, we're nervous about these flaviviruses. But I want to come back to the way that you generate the neurons. And what we've been finding is that if you use the same protocol across different lines, you end up with heterogeneity. Especially say with dual SMAD with nothing else added, we see this heterogeneity. And I think Barbara Treutlin's group has also defined differences when you use, say, Neurogenin 2, if you use that across different lines, or if you plate the cells at slightly different densities. So really my question is, when you look at neurons, how much difference is there between the neurons that are generated,
Yeah.
and I mean subtypes like layering, real fine differences between the lines, and how do you control for that so your village output is not so influenced by those?
Yeah. So I'll say a couple of things. We haven't really made a lot of post-mitotic neuron villages, but the ones we have made have been with a combination of NGN2 and dual SMAD, which we think helps a bit with the reducing that heterogeneity. What we found with our neuron villages, which are still kind of, I would say in the pilot, like maybe 20 to 25 donors, are not massive quite yet. What we found there is that timing appears to be the most important contributor to that variation, and that they seem to coalesce towards these sort of, I would say, just generic dorsal telencephalic cortical neurons by like day 21, 25 in that range. But prior to that, it does seem like our UMAP is showing a bunch of weird cell types. I don't want to say weird, just not a nice little ball, right? So I can't give too much insight into that quite yet. Like that data is like 2 weeks old. We're still trying to figure out what it exactly means. But we do know that it's a little bit better than if you just have NGN2 alone. I think that's what Barbara's paper was saying. Like if you just use NGN2 alone, you can get a range of different things, including like spinal cord neurons. And even in the Egan lab, before I left, they published a paper showing that if you use certain patterning factors, you can drive them towards a spinal identity just with NGN2 and those factors. So that's one thing I'll say. The second thing I'll say is with our neuroprogenitor cells, it is a much tighter ball, right? So when we look at the UMAP, they are much more similar to each other. Having said that, when you see on the UMAPs some of these extraneous cell types, we're identifying that a majority of those are neural crest. And so what we've worked on, we've spent pretty much most of this year, I would say, taking a pause on building villages and really focusing on cleaning that part up. And there's two things that we found. One, we switched to this AEVS1 safe harbor locus system because it allows us to do selection, pre-induction, and post-induction. And so we really have done a much better job of getting rid of the cell type contamination and only having the sort of a nice homogeneous neuroprogenitor cell culture. The second thing is we spent a lot of time optimizing our FGF. We found that was huge, right? So there's a lot on the market that were fine for, say, the first 2 or 3 passages. But then beyond that, we started seeing for some of our lines, pockets of neural crest that over time they took over the whole thing. So we ended up switching back to the original one that's in this publication, which unfortunately was incredibly expensive. but we're like, we're going to use this for now. And then we found one that's actually a lot cheaper from maybe Gibco. I can't remember the name of the company, which company came from. But if anybody has questions about which FGF to use, we will tell you exactly which one because we spent a lot of time with side by sides, and this one clearly won out in terms of purity of culture as well as cost.
Yeah, thank you. Yes, I think that it's really worthwhile putting a huge emphasis on these issues so that at the ground level, you're starting off with really consistent results. So thank you.
I'll just say really quickly, thank you, Sally, because it was something you said to me at a conference in Ventura, California, a year and a half ago, where I was like, you know what? If Sally's asking this question, we need to address it. And so we spent a lot of time with you in mind, actually.
Oh, very kind.
Looks like a DD has a question. DT has a question.
Hello. So I'm a PhD student in Helena Kilpinen's group. We're also working with some salvage approaches, actually, also using census-seq. So I had a different question initially, but now after this bit of discussion, I was curious, this kind of neural crest problem that you guys are seeing, do you see it more when you actually put the cells into villages, or do you also see it with the individual?
This is before we even make the villages. This was
Okay.
like this was like forcing us to cut 10 to 15% of our lines on village construction day because we realized they were just too dirty. And so for our villages that did have some neural crests, what we were doing is just bioinformatically removing them from our single cell RNA-seq data to make sure that we're focused on that particular cell type. But if you're talking about census-seq, that becomes a bigger problem, right? You can't bioinformatically remove the neural crest from census-seq. So luckily for the Zika stuff, it was a lot easier because that particular culture did not have these issues. I think it's because we were using the really expensive FGF at that time. It wasn't until we switched over here to UCLA and tried to cut some costs, and other labs were asking about SNAPS 1.0. When they started reporting these issues, that's where we're like, oh, maybe that FGF was actually incredibly important. We probably shouldn't have switched away from it.
Okay, well, I'll definitely follow up with you on the FGF in another conversation. But my original question was, I got really excited when you said you guys simplified the cell counting because I'm in the lab doing that and I know how long it takes. So what are you using now, if I can ask?
Yeah, it's I can send you all the details. Like the plate is the most important part, right? So I hope the sales agent is not going to look at this video, but we have what's called a citation that does automated imaging, right? And so we're always thinking like, how do we use this automated imager to count cells for us in a dish? The problem was when you put the cells in, like in suspension while you're trying to passage, they're not all on one layer. So they're kind of floating around. So we really were having trouble doing this. But then we looked into a company that was going to sell us a whole system for like another $80,000. And she told us like, oh, it's all based on this plate where you put these cells in like row A and they flow down into row C and they're now in a cover slip essentially. So they all are flattened out. It's just 10 microliters, all that stuff, right? So similar to the Countess slides. So once they're there, we're just throwing that into our citation. We developed some algorithms to help us identify the cells, which is pretty much what that company was going to try to sell us anyway, right? Like their special algorithms. I think it's CellPose is the software we use. We just train CellPose and then basically it takes 3 minutes to read through all 24 of those. And so that's what's really making this a lot easier. And we switched to the new rule in the lab is if you're doing more than 4 cell lines at a time, it's faster to just do the citation. If it's less than 4 or fewer than 4, just do the Countess because you're right there next to it. It's just, pretty quick.
Thank you so much.
Thank you. Looks like Ron has a question.
Jump in, Ron.
Yeah, it's neural crest. So there's a paper in Stem Cell Reports in November from people in Toronto. And as an aside, you'll note that people in Toronto think they're the world's expert on any kind of stem cell. But it's from Vanderkoy, from Derek Vanderkoy. And to just to whet your interest, basically he says that the neural crest is the ground state, the neural crest precursor. So it's not in his view, it wouldn't be an extraneous sort of contaminating cell. Now, you might argue with that, but the reason I made that sort of slight aside about people in Toronto is that you'd have to argue in an intellectual way, right? And so I think there's some very interesting topics that sit in the middle of this question about cell line diversity, which really might have to be addressed before one can get a systematic way of regressing it.
Right.
Yeah. Yeah. Anyway, have a look at this Van der Kolk paper. It's interesting.
You said it was stem cell reports?
Stem cell reports. Yeah. November.
Got it. I'll take a look.
Yeah. Well written, very authoritative. It's not necessarily accurate. That might depend on your training,
Got it.
but it's very interestingly written.
Good to know.
I have a question, and I seem to be struggling to raise my hands. Hi, Mike. It's
Hey,
Martine.
how's it going?
Good. Hi, everyone. I don't think I've met most of you. I'm used to work with Mike at the Broad. I'm now at UC Davis. And I think I'll be scheduled to present in the spring to talk about microglia.
Good to see you.
I had a question, Mike, about cell line selection being clearly in there right now, and even trying to focus on one disease. It's still like, yeah, like your different ancestry is still only 50 donors from each. So how do you take into account that all the donors have a different genetic background and different risks for so many diseases?
Yeah, early on, people were asking that question, and it kind of scared us away from doing diverse ancestries, because I think the prevailing idea was it's going to be, it's better to do 100 Europeans than 50 Europeans and 50 African Americans, what have you. But there's been a lot of work at the GWAS level showing how increased genetic diversity increases your chances of finding signal, whether it be a response QTL or NIQTL itself. So we don't know if 50 is going to be enough to find ancestry-specific variants of interest. But what we do know is that by having the mixed ancestries, it will increase our detection of these variants, assuming that this follows the same principles that we've been seeing in sort of large scale in vivo GWAS studies. So the idea is to make the village include all of them, and then bioinformatically run analysis on each group to see if there's any ancestry specific signal. But we don't know yet. It's a question, because we'll have to see.
Okay, and I assume since this was part of CERM, is CERM the most diverse biobank so far, or is there anything?
It's a good question. I don't know. Maybe it's like a West Coast bias. Actually, it's East Coast bias, because in the Broad we had 600 lines. So I kind of just thought that's all that was there, kind of growing up, right? It's nice here because at least for CERM, they like when you pitch using their resources.
Yeah.
And so there was a certain level of strategy involved in getting the funding to build it around CERM's resources. So I know they have, hundreds of diverse lines, which is really important for us. But I will say, those of you working with CERM lines, we just thawed 120 of them in the past 2 months. You really need to have XAV in the culture, which Rald has been doing that since day one. If you don't, a lot of these lines will differentiate within 2 or 3 days of being thawed. So that's just our standard practice now, at least for the first 2 passages out of thawing. They need to have XAV just to make sure that they can actually stay stem cells and survive.
Yeah, they're tricky, but I'm also biased for CERM. But thanks.
Yeah.
But you're growing them, just to continue this conversation, you're growing them as neural precursors. The cell you're proliferating is not the pluripotent cell.
Correct, but when
isn't that right?
Yeah, but when we receive them from CERM, they come as pluripotent stem cells. They come as IPS.
Right.
That we have
but just to understand, so the XAV, what the XAV is doing, I mean, I'm sort of saying this as a statement, but it's sort of a question. It's creating a telencephalic neural precursor proliferating population. Isn't that the cell that your SNAP cell?
Yeah, so we include XAV when we do the induction. I will be honest, I do not understand how XAV on stem cells, like XAV alone, how that is helping them maintain their pluripotency. But we have found that it really doesn't change much about them morphologically. And when we throw them into villages, they seem to be not becoming neuralized by the XAV treatment. To be clear, though, this is just for the wake when we get them from their biobank. Once we kind of expand these lines in-house, they tend to do a lot better. And so we don't have them in XAV leading up to induction. We just add the XAV again as part of the induction for 2 days. So I will say it doesn't make perfect sense to me right now. We actually recently adopted this for this new batch of SERM lines because our collaborator at the Broad, Walden MA, who handled 600 SERM lines, this is now her standard practice for maintaining them because they are quite tricky to behave. I don't know if that's a SERM issue or if other biobanks have this issue, but we're fully on board now with this because I think we would have lost half of these lines if we didn't start the XAV treatments.
I'm so glad to hear this because we have a set of SERM lines that we can't grow. They just diff. So
Yes.
we're going to try this.
I would make them in the XAV, if not
Great.
the day of, at least the next day. Start hitting. Yeah.
So maybe I can ask you guys another question. When they differentiate these lines, do they differentiate as gastroloids? Are they differentiating to posterior fates?
When we see them, I don't know exactly what they're becoming because we're just looking at the morphology. We can just tell they're not in colonies anymore, and they just become these weak, flat cells. And there's a lot of variation in the morphology of what they become from line to line. So we haven't actually looked into what is happening. We're just trying to stop it from happening as quickly as we could.
Very cool. Thank you.
Oh, you've been waiting. What you got?
Yeah, no problem. Indeed, related to what Sally said earlier, that we should care about what kind of different cell types and subtypes we might have in differentiation. I think in IPS, the subtypes we might have as a starting one might be also important, and XAV might somehow help in uniforming those starting IPS. So I think we need more data also in IPS from cell villages to understand the full differentiation issues we have. But I would like just to go back a bit to the neural crest, because we are having this issue, and we are having this issue especially in pools where when we have differentiated the same baseline lines separately, we don't have that many neural crest cells. And we call it neural crest cells, but to be honest, when we try to annotate those cell types, they are like pericytes. They have a strong identity for pericytes too. I don't know if you observe the same, if it's something specific to our protocol, which in our case is the Xie et al. It's like developing the LibC group. It's a 70-day protocol. So I'm quite surprised that so different protocols have the same issue. So it might be something.
Yeah, maybe you could send me like what markers you're using, because I will say that based on the single cell data, I think in our paper, we actually described these as like neural crest-like because they had some markers, but not all of them. And so we're just like, we're not going to spend too much time. I mean, it was like 5% of the village was that cell type in the paper. So we had to end up using just some sort of, I forget which protein marker we use for immunostating that is like the canonical marker for neural crest. That's what we end up using in our cultures to really see like, okay, are those cells that are popping up, are they most likely neural crest? And they were lighting up for that marker. But it might be different at the single cell level. So I'd be really interested in maybe we talk some more about what you're seeing, and we can say, are we seeing the exact same markers?
Okay. Yeah, it would be fantastic.
Because I didn't think about parasites, actually. I haven't even considered that as an option.
And indeed, for our protocol, it gets worse. So as long as the more time you have them in differentiation, the more parasites you end up having. And it starts with just 2 lines or 3 lines. And then by the end of the differentiation, most of the lines just have parasites, which is a problem.
Yeah. We will also say it's biobank specific. One of the things that really motivated our actions to try to optimize this was we got some lines from, I don't know if they came from Simon's Foundation. I don't know who made the lines, but there it was like 50% of the cultures, like 50% of those lines were becoming neural crests at like passage 3, which we had never seen it happen that quickly. And so we're like, okay, we got to go back to the drawing board because we have to make sure this protocol, which was optimized for all the embryonic stem cell lines and a bunch of SERM lines, there clearly are biobank specific challenges. And so we're trying to make our protocol robust across everything you can find globally.
Yeah. This is such a helpful conversation of how we just are doing an experiment with Dan Geschwin's lab of really deeply phenotyping over 100 IPS lines. I know it's a small subset, but they're the lines that we work on a lot, the MAPT mutation, the controls that are relevant. We're heterogeneity at the IPS stage. The FGF2 concentration is absolutely critical to keep them the most pluripotent, to get the best differentiation. But I so agree with you that we don't understand the heterogeneity of iPS cells, what really makes a sort of quintessential, reliable one, and sort of how to control for that process. And, it's really great to share notes like this. I appreciate it.
Yeah, definitely agree.
If I can just add something to this conversation, it's not from like personal experience, but I heard a talk from Madeline Lancaster recently where she actually talked about something similar about IPS heterogeneity. And for them, the Wnt inhibition, which is what ZAV is doing, the XAV, worked in many lines. But what really worked for them was they realized that it was this kind of aberrant posteriorization of their IPS lines that was leading to poor differentiations. So they just wiped clean the epigenome with like kind of a cocktail of epigenetic modifiers. So they wiped it clean, let the IPS rebuild their epigenetic marks, their landscapes, and then they differentiated way better also in like kind of mosaic organoids.
Yeah, I think she just this past week, Sally, isn't that what she talked about as well, the cleanup? That's a cool thing to bring up.
Yeah.
Yes, it is. We also hesitate, right? What's all of that? Because there were a ton of chemicals
Yeah,
in
yeah.
that.
It was a smoothie. It was not a yeah, it was a whole lot of stuff.
Yeah, it sounds kind of terrifying.
Yeah, awesome. Yeah, although neural crest, if they are looking parasite-like, it could be that you've got Schwann cell precursors in there because it's a shared
Yeah.
progenitor. So there are markers that you might look at related to that lineage.
Great to listen. Thanks.
Yeah.
Great, guys. It's great that the conversation after is so useful. I think it's good to hear you guys swapping extremely precise pieces of information that make big differences when you're gonna wake
Yeah.
up a whole new set of lines.
Yeah. I wanted to talk to Michael about FGF2. We can do it another time, but
Sure.
we've spent a long time looking at FGF2. And, the half-life is 4 hours. Therefore, we made a sustained release compound that we add. It's available to keep the level around 10 nanogram per mil, no matter what, because some lines are very sensitive to a drop of FGF2. And what we found through this whole process is that different companies have different amino acid sequence that they all call FGF2. And you have to be very careful where you order from, what QC they do, and just, be careful. But if you want to try the sustained release FGF2, we can send some to you. We found that it improves the pluripotency. And so that all of the work that we've done with Dan Geschwind includes that during the growth of the iPS cells.
And that's in your Bertucci et al. Preprint, right? The whole FGF
Yes.
and dual SMAD dosing. Yeah.
Yeah.
Just so people know it's out there if they want to.
Yeah, and the other thing that we've done with Dan is compare the same lines grown in different media. And if you look at StemFX and M-teaser and M-teaser plus, for example, they have very different transcriptional profiles. And they're done side by side. And that tells you that these pluripotency media are really different and they're affecting the cells differently. And, we often think of them as just being equivalent, but they're really not. So, yeah. It's a
What
lot.
I can do is I'll have my grad student put together all the data from those trials of side-by-side comparisons. And I'll also have him track down, like for each of them, what were the amino acids, what were the products, all that stuff. We can assemble all that for you, and we can
Purity. Oh, awesome. And then maybe we can chat.
exactly. Yeah.
Anyone who likes FGF2 conversation.
Maybe if we're having such a detailed conversation about culture components, I could ask you just open-ended question about laminins.
Mm.
Have you guys looked at laminins and different laminin isoforms?
I don't
No.
think we use.
You
You mean
you
like?
talked about integrins in your talk.
Oh yeah, as the entry factors for Zika.
Right.
Yeah, we did some follow up on the integrins to show that they were, and others had published that around the same time. We I remember like having that, making that discovery, and then like 2 days later seeing two papers come out and being scooped on the same day twice. I'm like, oh well, looks like I was right. I was super late in the game. But yeah, that I haven't looked at any laminins.
Oh. Again, there's another recent paper from the Karolinska group doing a really detailed analysis of particular laminin isoforms. And again, I mean, I think what people don't really understand is how quickly these cells make computations.
Mhm.
So there they have a really beautiful Wnt dose response curve, for example. And again, I just sort of draw it to our attention. I mean, maybe we need to sort of create another lecture series where those of us who are really interested in this go into all the details at exhausting length. Yeah,
So Ron,
great.
who is with me, has done a lot of work on winds previous times, right, in Cardiff, and looking at the impact on the patterning of NPCs. So yeah,
Oh, cool. We should talk more.
yeah, be great. Really cool. So I'm just so excited, Michael, about your approach and what you're accomplishing. It's really outstanding.
Thank you. Yeah,
So excited.
looking forward to keeping this conversation going with you, and so we can keep improving upon everything and making it as robust as possible and as useful as possible.
So important. Yeah. Thank you. Thanks, everyone.
Yeah. Thanks,
Thank-
Michael, and great discussion, guys. Good to see us connecting on so many levels.
Bye, everybody.
Well,
Bye. Bye-bye.